The first, I can't remember how many questions. Is it 6 questions make up the, or is it 5? 1, 2, 3, 4, 5 questions then make up the past section. So really important that you've got the hang of the past material, and then anything else actually, just develops that really. It is possible to contribute towards merit and distinction grades within the past section, but you won't get a full merit or distinction grade from the past. But the quality of your answers in that first section can help to tip the balance as you go into the second section. So it's always worth being fairly thorough about the answers. Okay, first of the questions then is about the gross structural features of, and this is the nervous system, so it's explaining the roles of features, and so it's about relating structure to function. And so we have a diagram of an eye with various bits identified on there, and when you get diagrams like this, you might not have time, but and I can't remember quite how I've structured the question in the exam, but I haven't here asked for names of parts. I think in the real exam I probably have, but I would be tempted to sort of start with just getting my bearings around it and perhaps naming the parts on the diagram. I'm not, however, going to do that because time isn't on our side really for it, and I'm going to just go through the parts that are identified in the question. So the first bit, describe the cells found in part F, so that's this little bit here, compared to the rest of the retina. What do we call part F? That's the fovea. And we have 2 types of cells in the retina generally. Which type are found concentrated in the fovea? That's the cones. And there are very, very few cones as far as the rest of the retina is concerned. So that's really what that's about. And that's the part of the retina where we focus images that we really want to concentrate on. So although I haven't asked for the name, actually it's not a high concentration of cones, it's only cones, isn't it there? So cones only. And then it asks for a comparison. The rest is mostly, or perhaps not mostly, a sort of higher number of the other type of cell, which is rods. That's right. Okay, so that's a comparison. Okay, describe the cells, they're cones only. I haven't left a space for you to actually describe them. So, I'm not needing you to sort of describe the structure of the individual cells or anything like that. So it's just describing them compared to the rest of the retina. Right, coming down a little bit, we have, how does part A help when focusing on a close object? What's part A? Yeah, ciliary body containing muscle, yeah? All right, so we could call it ciliary muscle. So I'll put body stroke muscle. And so what does it do for focusing on a close object? Will it adjust the lens by contracting? Yeah, which one for close? It becomes more curved, so it actually contracts for that, yeah? So it contracts. So the lens actually relaxes then, doesn't it? It's not stretched out. It's to a thicker shape. I think that's the right way around. The trouble is that requires you to understand the physics of light, doesn't it? Bending light, refraction of light, and the shapes of lenses generally. And yeah, we don't actually cover very much of that. Okay. So that was part A covered. What's the main role of part D? What is D? The cornea, that's right. So D is the cornea. And its main role is, it does let the light in, but it is part of the focus. So it's refraction of light, refraction being the bending. Sorry? D is the cornea, and it actually lets the light in and bends it. Front bit is the cornea. Yeah? It's the front part of the sclera. Sclera is the bit. Yeah, yeah. And then so the sclera is protecting it's holding the eyeball in shape. And then D, the cornea, is the little window in the front, and light passes through that, and as it does so, it's bent on its way through. Because that's the bit that's then adjusted with laser surgery to correct people's eyesight. Right, so that is the main source of refraction. People think it's the lens, but the lens just allows alteration of the focus, really. The cornea is the main refracting structure. All right, so that's part D. Now, part C is the pupil, if you're talking about, yeah? Part C is the actual little window, yeah, there. And that's the problem when you look at it sideways on. You look at that from the front, and that's the little black hole in the middle, isn't it? See? Yeah? Right, what we got there? Now, part G is the blind spot. What structure leaves the eye at this point? It is the optic nerve. So this over here would be the optic nerve. So we haven't got any light-sensitive cells there. No, the structure is called the optic nerve. That's the bit that connects it to the brain. And then which part contains a black pigment that prevents internal reflection of light? Well, you could give it its letter if you wanted, which is H, and it's called the choroid, yeah. Okay, that's the first bit done. Now, I've asked you for 1, 2, 3, 4, 5 bits there, and probably if you got 3 of them out of that 5, I'd be happy with that. You've just got 1, okay. It's obviously a bit of work needs to be done on that. Right. The second part of this question then is section through the brain. And we've got a table down here with names and functions, some of which I've given you, others to be filled in. So we'll be up and down a bit here. So part A is a band of nerve fibres that connect the right and left sides, and it's called? Oh, we've got a nice chorus of corpus callosum. Oh, great. Yeah, that's the problem with tables on the smartboard. And I've had that happen before. But you get it. You've heard me say it, you can write it down. Right, so that's part A dealt with. Now, part B, the hypothalamus. And you can keep this quite simple. You don't need great elaborate sort of descriptions of it. But this bit here, all right, is the hypothalamus, and it's not a part that we've sort of described in a lot of detail, but any offers for its function? Hunger and thirst, and it connects with the pituitary, yeah, automatic sort of stuff. It's very much a halfway house between the nervous system and the endocrine. So it's almost like a monitoring centre for things like hunger and thirst. So hunger, oh, here we go again. It's jumping about all over the place. Oh, no, it doesn't want to do that with the rest. And, oh dear, connects. That's by no means all it does. the other thing, it does produce hormones, yes. That's part of the pituitary link. Monitors body temperature. I should have put in there. Hunger first. Right, we'll use that little space. Temperature. And the pituitary. That's interesting, isn't it? I only wanted to move the H on there. So hunger first, temperature. So yeah, monitoring body temperature. That was the only bit in that thermoregulation section that we took out of the handouts that would actually be any use, really. So yeah. Okay, part C is the cerebrum, main function. That's the stuff, yeah. So we've got sensory input. Oh, this could be all over the place. Motor output. And then it's happy once it's done that, isn't it? And then it's mental functions. Yeah, I'm not going to try and write. We're moving down this time.
Mental fun.
Mental fun. Oh dear. I'm going to have to write it outside then. Huh. Okay. That is very strange. That's really odd because on the previous one the H jumped upwards, but on this one the SE stayed down and the rest of it jumped upwards. I have no idea what's going on, but never mind. Right, part D coordinates voluntary movement and balance. We are going to call that the cerebellum. Oh, let's jump down and up a bit. Okay. And then part E, the medulla oblongata. Yeah, that's it. Automatic. Whoa. Functions. Oh, this is so hard to write like this. E.g., I hope I haven't got any more like this. Heart and breathing. Oh, great. We have got another one coming up. We will, yeah, I'll find a way, don't worry. Obviously, you have tables on your exam papers which don't do that.
Are they 5.6 then?
What do you mean? The sort of, that's just, yeah, but it's sort of, yeah, I'd be looking for more than half on each table, basically, to pass on this. So, you know, if you got 3 or more on both tables, you would pass. If you got 3 on one and 2 on the other, I would be dubious about passing you. But that is still half of the whole lot. It depends how wrong you get it, I think. You know, in terms of parts to be labelled, if you name it, give it a name of something that's sort of similar or in the same area, and I could see there was a misunderstanding, then I might be more generous. And if your descriptions were a bit perhaps light in terms of accuracy, you know, it's very hard to say, but you're looking for, you know, getting more than half on each of the subjects there. But you don't have to get absolutely every one right. All right? So I do have to be, you know, reasonable about it, but also have to be sure that you've actually met the assessment criterion. And that's not always by knowing everything. All right, so that's the first of the assessment criteria out of the way. Now, the next part then, this, hopefully this won't jump about. We've just got the gap fill approach to this, and if you can pretty much memorise this, then you're in with a fairly good chance. So let's start with the resting potential then. The sodium-potassium pump delivers 3 what ions? Sodium, so you can either put sodium or you can write Na+, that's fine. All right, if you've put both, even better. And you see, that's the sort of thing where if you lose one somewhere else, I might take account of extra information that you've put in. So they're going out of the neuron, and 2 K+ or potassium ions, yeah, going into the neuron by the process of active transport. Certainly is. Okay, so that's the bit that requires energy because it's moving them against the gradients. So this results in concentration gradients for sodium and potassium, which favour the, it is diffusion. If you just put the word movement there, actually that would be okay, but diffusion is preferable. All right, so, you know, if you just got open channels, they would both then be diffusing back out where they came from. So they favour the diffusion of the ions in the opposite direction. However, the process involved is facilitated diffusion involving ion channels is the word. And this goes back to the cells unit. So this is, you know, a particular problem for charged particles to get through the membrane. So they have these special ion channels. These ion channels, they are, I think they're voltage gated. So the voltage difference in the membrane will open and close them. Anyway, at this point, the sodium channels are mostly closed, but some of the potassium channels are open. That's it. Consequently, potassium ions leak back out of the neuron, while sodium ions cannot pass back in. So this results in an accumulation of positive ions. It's positive ions something the neuron compared with the two parts of the neuron you could compare are outside and inside. So the positive ones accumulate outside compared with the inside. Now, that's not to say that we've then got a real sort of positive or negative charge, but this is a comparison there. The membrane is described as being polarized, so I haven't asked you to remember that word, you know, like several of the others, but you need to know it. All the content of this you need. I will put different gaps in the exam, all right? And the resting potential has a value of approximately -70. Whoops, all right, so that's describing the inside compared to the outside. Okay, so that's the resting potential. Then, in order to actually make use of a neuron, we need an impulse, and the impulse consists of the action potential moving along the neuron. And so it has to start with a stimulus to or beyond the threshold level. That's something that we don't look into in detail. You can do, but it's not part of what I do. But when we have that stimulus, which channels open? It is the sodium channels. So it allows those ions to rush into the neuron. Increasing numbers of these channels open by a process of positive feedback. It's that rare example of positive feedback, resulting in the inside of the cell becoming positive compared to the outside. So you've got here, yes, you have to use the same word, well, or more positive if you like, but becoming positive compared to the outside. The membrane's described as, it is depolarized, isn't it? And that's one of the difficulties in understanding this, because that makes it sound like you've then not got a difference between inside and outside. And the action potential has a value of approximately +40. Yeah, if anybody puts 50, you'll still get it. Almost immediately, these channels close and the potassium channels open. I've clearly left enough space to write potassium. Allowing these ions to rush back out, and this repolarizes the membrane. Well, it repolarizes it to the point of hyperpolarizing. It repolarizes first. You get that little graph where it goes like that. That's your resting potential. Then it depolarizes, it repolarizes, and then it hyperpolarizes. So the hyperpolarization is there. All right, so that's depolarizing, that's repolarizing, and then that bit down at the bottom is the hyperpolarizing. So it's the tail end of the repolarizing. I wouldn't mark it wrong. All right, it would depend what else you'd written in there as to whether I felt that was, oh, well, that's all right, you'd be fine on that, yeah. if that's the only thing, because you've clearly understood what happens. But yeah, the repolarizing is the key bit first. Back down to this point, it's restoring the minus 70, and then it overshoots a bit, and that's the hyperpolarizing. So far, so good. All right. Now, the questions on the synapse here. Right. Which of the following ions enters the axon terminal when an impulse arrives at a synapse?
Calcium.
It is calcium. And which of the following are neurotransmitters? Is acetylcholine a neurotransmitter? It is, yeah. We mentioned just a few. There were about 4 I mentioned, but acetylcholine is probably the best known one. It's the most common one in the whole nervous system. And we talk about acetylcholine esterase as being the enzyme that removes it and that sort of thing. So it came up sometimes just abbreviated to that, if you see that anywhere. Right, is noradrenaline a neurotransmitter?
Yeah.
It is. Is insulin?
No.
Is adrenaline?
Yes.
Adrenaline's not a neurotransmitter, really. It's a hormone. Noradrenaline is the neurotransmitter version of it. All right, so adrenaline is a hormone that travels in the blood, isn't it? Yeah? But noradrenaline in the sympathetic nervous system works in the same way as adrenaline, the hormone. All right, and this is where you'll have come across them in your assignment, adrenaline and noradrenaline, or please don't write epinephrine and norepinephrine because that's the American version. All right. But yeah, the epinephrine or adrenaline is the hormone, and the nor version is the nervous system version of it. But that, again, it's one of those areas of sort of overlap between the two. But adrenaline is a chemical produced by the adrenal glands in response to stress, and it's a long-lasting hormone. So I wouldn't classify it as a neurotransmitter. What about dopamine, though?
Yeah.
Yes, that is. I should probably make sure I don't. Oh, I can't now. The exam's in the exams office. I should make sure adrenaline's not on there because of confusion. Okay. What happens to a neurotransmitter after it's been released from the presynaptic cell? First of all, no, first thing is it's got to bind to the next cell. All right, it's not removed straight away. So yeah. Okay, so crosses synaptic cleft. Yeah, some people have an advantage of sort of having met it before. Second thing is that it binds to receptors on what's best described as the postsynaptic cell. You could just call it the second cell if you want to. Thirdly, that opens up what sort of channel? Sodium channels open because we want to depolarize this.
I said it triggers it to depolarize.
Yeah, I mean, I haven't actually. That's not really what happens to the neurotransmitter, is it? So we can forget that thereafter. I've gone too far. I'm not answering the question. The neurotransmitter is then removed by enzymes. That's it. So the neurotransmitter removed by enzymes, and it's actually going to be taken back up into the presynaptic cell so you can reuse it. Otherwise, it's a real waste, isn't it? So, but an example there would be, just to reinforce the name of this one, acetylcholine esterase. I have a feeling we met that on the substances that can interfere with synaptic transmission. I can't remember which one it was that interferes with that. But it might also be a target for some of the anti-Alzheimer drugs. because it will then raise the level of acetylcholine in the brain and improve the function of synapses. So I have a feeling that, yeah, there's an Alzheimer treatment that targets acetylcholinesterase. That's the key, really, to the answer to that question. Yes, it does its job, but then it has to be removed and then taken back into the presynaptic cell. Now, again, for that whole assessment criterion then, you know, you've got quite a lot of answers, and I would be looking for correct bits in each section, really. So you might get the resting potential perfect, the action potential completely wrong, and bits of this right. You're not going to pass, all right, because you've not got anything in the action potential bit. So if you look at the actual assessment criterion is described aspects of the cellular mechanisms involved in, and then it's nervous response, obviously. So the mechanisms are, you know, what sets up the resting potential, what's an impulse made of, and that's the action potential. I've not included in here a lot of things I could, like the fact that this action potential is an all or nothing response. It jumps from gap to gap in the myelin. I used to have questions on there about the factors that affect the rate of transmission, like the diameter of the neurons and myelination and that sort of thing, but that's not in there at the moment, I don't think. Oh, is it? Oh, that's where I've seen it. I've put one of those in. With the three animals? The cat, the lizard and something else? No? Oh, it's a squid, is it? Oh, right, okay. Yeah, that... It's amazing that the obvious comments that are made in questions sometimes, and that can put you off, all right? Because obvious things, you think, are they trying to trick me then? You know, I know that's really obvious, but yeah. All right, so again, I'm looking for sort of key parts, I suppose, of each section in there, but the actual exam paper's not quite like that. Okay, now 2.1 circulatory system. Is 2.1 the circulatory system? Probably. I'm never sure whether I've got circulatory and respiratory the right way around, but it doesn't really matter, does it? Okay, 3 components of blood plasma. The most important one is water. And then what else is... the proteins are definitely important. Ions, yes. Do you want to throw something else in for good measure? No, clearly. Hormone, yeah, we could have hormones. And we've missed things like nutrients, oxygen, and waste. There is, I suppose, not a lot of oxygen. Let's remove that one. It depends whereabouts in the body you are. You're not going to get it in most parts. Let's put nutrients and waste. I don't want to confuse matters too much. The only way for oxygen to get in and out of red blood cells is to pass through plasma. So you will find oxygen in plasma. But I suppose it's not significant enough, really, to be in there. So we will keep it more obvious. Okay, I've left a lot of space for this next bit. The role of B lymphocytes. Well, first of all, they have to recognize something. Antigens. So it's the recognition of antigens. So they recognize antigens. Now, those might be on the surface of the bacteria and viruses. They might be toxins, for example, that are released by bacteria. So you've got surface, and you've got toxins. So it's that recognition bit. And then B lymphocytes, well, they produce 2 things. Antibodies. What else do they produce? What do all lymphocytes, well, what do all the white blood cells, well, what's the important bit about retaining immunity? Memory cells, yes. So if we were then unlucky enough to meet the same bacterial virus another time, then hopefully, we might have retained some memory cells and wouldn't be infected again. Obviously, that doesn't always happen. We get these vague infections sometimes, don't we? Let's say, somebody might have, I don't know, food poisoning even, or something like that. You don't retain a memory of that. So if you had salmonella once, you don't then become immune to it, do you? Otherwise, you could have salmonella once and then eat all sorts of, disgusting stuff. But we don't have an immunity to that. And that is essentially an infection. But the general sort of response of B lymphocytes then is the production of antibodies that will help to remove the antigen-bearing substance, and the production of memory cells for response at a later time. And you can, of course, just be given antibodies, and they won't give you immunity because there's no production of memory cells. Yeah, that, not surprisingly, is an important part of the answer in the exam. Right, keeping it fairly simple here, you can add quite a lot to this. Right, 3 features of erythrocytes. Erythrocytes are otherwise known as, these are red blood cells, that adapt them to transport of oxygen. Well, there's loads more than 3 features, so what should we go for? Okay, so no nucleus. And why is that useful? That is right, there's more room for hemoglobin. So a second feature could be, well, I was going to say they contain hemoglobin. Can I just put contain hemoglobin in there? But yes, I've now got surface area and shape coming up, which are, so they contain hemoglobin. I'll come back to the reason why that's useful in a minute. So shape, it's this biconcave disc. And so this gives you the large surface area to volume ratio. Now, the reason that hemoglobin is useful, apart from the fact that it's able to pick oxygen up at high partial pressures, let it go at low partial pressures, what the hemoglobin is actually doing is maintaining a concentration gradient, because it's actually removing oxygen from the blood, as it were. So that means that when the blood cells come round to the lungs, oxygen needs to diffuse down a concentration gradient from the air into the blood. So you need it to stay high in the lungs but low in the blood. So as soon as it gets into the blood, it'll pass through the plasma into the red blood cell, be picked up by hemoglobin, and that's like hiding it, so it's no longer evident. It doesn't form part of the concentration gradient. So apart from the fact that it's a useful sort of transport mechanism, it also, I think, oh, I can't actually fit that in there. Well, as I wanted to say, it maintains the concentration gradient. You'd think it's not going to apply at the other end, but because you've got different processes going on at the other end, so maintains concentration. I'm just going to put Konkan gradient, because Konkan gradient. Konkan, like that, CONCN, always used to be a standard abbreviation for concentration. It sounds like the laptop trolley coming down the corridor. All right, okay. Something else coming down the corridor. Function of platelets, basically. They initiate clotting, that's all we need on that one. You could have had other things with the red blood cells, of course. Size. The small size increases the large surface area to volume ratio. And so that's the first bit of the circulatory. Circulatory is quite substantial because you've got blood vessels, and you've got heart. So, you know, several parts that you need to be okay on here. Right, complete the labels on this diagram. I've put rather a lot on here. You don't need all of this on the exam one. Where shall we start sensibly? I'm going to start with the top right and go anti-clockwise so that I gradually move that way across the diagram, so I'm not then standing in front of it. All right, teacher technique. All right, if we start with this one here then, what's that? That is a pulmonary vein. Coming back in on the left side of the heart from the lungs. Next label round is left atrium, followed by bicuspid, yeah, bicuspid or mitral. And then round at the bottom we've got left ventricle. Coming round the bottom there we've got right ventricle. Coming up we've then got the tricuspid. Followed by this one here. Yeah, that's the inferior. I almost missed that one actually. This is way more labels than you need, but never mind. Next one up, right atrium, yep. Next one is the superior, yep. Then we've got the pulmonary artery, yeah, that's a curious one. I've not seen one that divides quite like that on a diagram before. So pulmonary artery, and up at the top we have aorta. Yay, that's that lot identified. Yeah, yeah, quite a lot to catch on there. This valve here they've labelled as a semilunar valve. That would be the pulmonary valve, and then the aortic valve, of course, is going to be up here behind that crossover bit, so it's going to be at the base of the aorta. So that's the bits sorted out. Now, describe the path taken by the wave of depolarization which passes through the myocardium during a single cardiac cycle. All right, don't make it too complicated. Where does it start? Sinoatrial node, yeah. Okay, from there it spreads across the, well, atria generally you can have. While it's doing that, it hits the AV node, yeah. From there it goes down the, yeah, into the, yeah. And then the wave passes, yeah, the Purkinje tissue is, which I've gone for now, Purkinje, all right, is part of the bundle of his, but it's then spreading, well, it's up the walls of the ventricles, isn't it? So up the ventricles. Concentrate on that particularly. All right, there's a big hint, the sinoatrial node. It is the point at which the contraction starts. It's the point at which you've then got the potential to modify the rate of contraction. And any nerves that are attached to the heart are going to be attached there. I'm not sure that's the only place. There are nerve fibres running through the heart, but you know. if I was to set a question like that, then yes, a chart, a sequence like that is fine. Yeah, you don't have to write everything out in full sentences. All right, I'm looking for keywords for links. If abbreviations, you have to be a little bit careful with. But yes, if you want to put something leads to something, an arrow can sometimes say that. Or if you want to say something increases, you can use an up arrow, or decreases, a down arrow is fine. So, if you want to talk about increased heart rate, you just put arrow up, heart rate. And that's okay. All right. So, yes, you can find ways of saving a bit of writing time, at least. All right, for blood vessels, this is just identifying the differences really between arteries, veins, capillaries. The exam, it's really arteries, veins, but we need to weed the capillaries out. So which is which here then? Which ones are carrying blood from the heart towards the tissues? Arteries. Carrying blood at high pressure? Arteries. Have walls only one cell thick? C for capillaries. Valves to main one-way flow? V for veins. Thick muscular walls is A for arteries. Exchange between blood and tissues? That's our capillaries. With one exception, carry oxygenated blood? Arteries, yeah. Blood back to the heart from the tissues? V. And blood is pulsatile? A. Is that the last one? I've managed to fit them all on. That's neat. All right, so you get a table to complete on that, on the exam. All right, to write the comparisons. And the better your written comparisons, the better you're contributing to your grading. Because I have had, so it's a question I use quite a lot. So let's say, description of the walls. Well, I've had people put for the arteries bit thick, for the veins thin, and leave it at that. And yeah, that might pass, but with arteries, it's thick with a lot of muscular and elastic tissue. For veins, it's thinner with less muscular and elastic tissue. And when you're looking at what's contributing to the higher grades, it's that sort of style of answering. But if you can only remember thick or thin, then that's fine, because that'll get you the pass. So just yeah, be careful. Right now. This question then about the formation, composition and function of tissue fluid. I've actually given you a word list here, which is a bit kind, although sometimes can be. No, it doesn't always. Yeah, it doesn't always work for everybody. Quite likely, yeah. Yeah. I think, although I'd said using all the words. Each word from the list once, yeah. Okay. Right, blood entering the capillary network, yeah. So that's that one gone. In the tissues is under high pressure, as it has come from arteries. Oh, it doesn't look like I made much effort when I was putting the word list up. The walls of the capillaries are permeable. So substances can easily be squeezed through them. Tissue fluid is forced out into the surrounding cells. I haven't written that very well. It contains small molecules such as water. What else? Oxygen and nutrients, yeah. Okay, so water, oxygen and nutrients. Water, nutrients, oxygen. But does not contain or, that's it, protein or cells. Too big. And that's a useful overlap with the kidney as well, of course. So protein and cells. Oh, I don't think there is. Is there a spare one? No. Is there? Oh, right, yes, there is, yeah. Tissue fluid supplies material to the tissues, removes waste, which is returned to the blood, leaving capillary network in venules. And then, yes, you've just got to creatively stick the word low somewhere. Whoa! Right, yeah, so we're left with that one. I wouldn't normally do that in a real exam, so yeah, don't panic. Okay. Yeah? Yeah, I mean, different styles of questions suit different people. And what you can do, of course, if you find a style of question that suits you, you can write your own questions. As you're revising, write questions in that style that will help you learn it. All right, you're not going to get them all like that in the exam. But, you know, because the learning's the important bit, then, yeah. That's anything that will help, really. Okay, respiratory system. This one should have been very straightforward. This is really, I would say, not beyond GCSE. So, we're only looking for approximate values here, really, but nitrogen in, 78 to 9, and nitrogen out, yeah, let's call it 78 today. And the reason for that, yep, and body doesn't produce it either. Not used or produced. Right, not used or produced. So what is it that lurks about at 21% then? It's oxygen. And when we breathe out, it's roughly, okay, 16 and 17%. And the reason it drops is, yeah, I'd say enters the blood, because what we're looking at is why is the air different. So it leaves the air and enters the blood. So air to blood. Oh, all right, that's where it's gone. That's explaining the difference. So not surprisingly, the next one is carbon dioxide. Goes in at, yeah. I shouldn't have put percentages here and here, because they're already up here. Table jumped. So why has it gone up? Using the same approach that I had in last. Yep, so it's moved from the blood into the air. Yeah, that's the key that we're looking at. Why is it changed in the air? Well, it's been put into the air by the blood. Yes, you can say it's a waste product, and as you know, as I said before, the more you add into your descriptions, the more good it does you. Then water is always a slightly odd one. Inhaled air, variable, and then out. It is saturated, yeah. It might be saturated on the way in, but reason. It's not so much. No, it's not so much to do with the carriage of gases, but just the surface of the lungs is moist. Haha, we got the word in. Lung surface is moist, so you just get water just getting into the air from there. All right, now the next bit, what are the structural features of any efficient gas exchange surface? A large surface area, you've got to think Fick's law, haven't you? Yeah, thin will be the next one. That's it, so large concentration gradient. Yeah, we could put moist in again if you like, yeah. We just, the presence of water will make it easier for the gases to move, so we can add that on as well. Yeah. All right, so we've got a good... You could put damp if you prefer, or covered in a thin layer of water. All right, now those three, of course, you've got the Fick's law approach to it, and it's just worth remembering that. Okay, describe the process of breathing in. First thing that happens, so intercostal muscles. Not sure I've got room for all of this. Contract. So the ribs move. Whoops. That's it. So ribs up and out. Then alongside that, what? Yep. So diaphragm. That's it. That's a good way of putting it. Contracts and flattens. So yeah, that's a down movement. So what would be the next thing that we put in? Volume, yes. Volume increases. So pressure. V1, V1 and all that, of course, decreases. So we've now got a pressure gradient and air moves in. Yeah. Once you've got all this, this will form the basis of good material for revision notes. Right, does that leave us with the kidney? Okay, now this bit then. Let's see if I can fit this on the screen a bit better. All right, I'm going to do this in more of a logical sequence, I think. If I start the beginning bit of it, if you like, here. Yeah. And as far as function's concerned, what's the main process we've got here? I would just put ultrafiltration for now. You can add an explanation if you want to, but that's, just putting that is really the main process. It's a good word. The next bit. All right. Okay, so proximal convoluted tubule. What's the main thing going on in there? Yep, it's selective reabsorption. We don't just take everything back. But yeah, you might like to consider what's taken back there. I mean, particularly glucose, quite a bit of water, some ions. Yeah, yeah. Yeah, I think they go back there, don't they? I think I'd managed to avoid mentioning them, to be honest. I'm going to miss out the next one and just go down to the bottom one for now. Loop of Henle. So I couldn't. How do you make an arrow go to the whole thing? Yeah, but then it would be ascending and descending limbs. That's why. Well, that was the reason I put the arrow at the bottom. It depends. Had you put descending limb of loop of Henle? If you just put descending limb, I can't mark it wrong. In that case, yeah, I'd have to mark that right, really. Yeah, if I was doing the descending limb, I would probably have put my label line to here. You know, that would be the descending limb, and that would be over the other side the ascending. Yeah, but I put it down at the bottom to try and make it a little bit more obviously the whole thing. Clearly didn't work, so that's fine. It's all right. It does help me see. You know, it will help me with my marking. You know, if I can sort of see where any misunderstandings might be. But generally, the function of the loop of Henle. Well, it's reabsorption of water, isn't it? So is the real key there. It's movement of sodium and chloride ions out of one side so that water will come out of the other side. But the main point of the loop of Henle is to get water back into the blood, because if we let it all go, we dehydrate. And that's the bit that varies in other animals. So if you know, desert living animals will have a very long loop to get up a bigger countercurrent multiplier and all that. And if you put countercurrent multiplier in there, it would help. You know, I mean, I'm keeping these very brief functions. The other bits that we've got, then if we carry on with our filtrate, it ends up here in the collecting duct. Function. Well, yeah, drains urine right to the pelvis of the kidney. It's also involved in water control, isn't it? Osmoregulation. Because that's where the next bit comes in, the effect of ADH and the aquaporins and all that. And that leaves me with, over the other side it is?
Renal artery.
It is the renal artery, yes. If you follow it around, you'll find it connects up with the glomerulus, so therefore, absolutely right, yeah, so delivers. All right. And then, how have I done this bit? I couldn't remember what I did with this. Explained the process of osmoregulation. So this has now got the two things that we met that are involved in osmoregulation. So you've got the loop of Henle and its countercurrent multiplier. Oh, I've just answered the first bit. And then you've got the ADH, the aquaporins, and collecting ducts bit as well. Because they are, osmoregulation as far as the loop's concerned is just a continuous ongoing process, and then the other side, the ADH production, is, you know, dealing with fluctuations. So which part of the nephron sets up this countercurrent exchange mechanism? That's the loop of Henle. I didn't call it a countercurrent multiplier there, but it's, you know, same. It's the countercurrent bit that's important. Right, what's the descending limb permeable to? Water, yeah. Right, but not ions, whereas, oh dear, the ascending limb is permeable to? Yeah, particularly that and that. That's the first bit. You know, I've not really sort of gone into asking questions, but those sodium and chloride ions leave the ascending limb. They end up in the space sort of around, well, the fluid around the loop generally. That then draws water out of the descending limb, so the water will end up going back into the blood. Okay, now, where are osmoreceptors located? Yeah, we could have, hypothalamus could have got mentioned earlier, couldn't it? Osmoregulation. Yeah? Just as I read that, I thought, oh, when we were looking to do functions of the hypothalamus, although it was involved in hormones, so yeah. But it is part of that, regulating and recognizing thirst, for example. You know, when osmoreceptors detect the blood's rather concentrated, that's actually going to, you know, one of the things it will do is make you drink more. So, which hormones released when when the osmoreceptors detect low water potential? Certainly is ADH. And the effect of that is, yep, more aquaporins in the collecting ducts. So that's right, more water returns to the blood. Okay, that antidiuretic effect means that it's not lost in urine, it's retained in the blood. Remembering, you know, ADH, of course, also known as vasopressin. We don't call it that these days, but I just take that opportunity for a sidetrack into, you know, remember if you put more water back in the blood, of course it will raise blood pressure. So that's why ADH does have an impact on blood pressure control, that it can actually raise the volume of fluid in the blood. All right, so that is the past questions.